heartiest welcome to all of you. Myself Nitish Kumar. I am Assistant Professor and Neuroscience Technologist from Department of Neuroscience and Technology, Yenepoya Allied Health Science College. Today we have a very interesting topic in front, like as you can see, it is ontogeny of central nervous system. So we will be studying the development of nervous system, the development of brain, development of spinal cord throughout the session. So we'll be studying it under the embryological brain development. We'll be studying the spinal cord development. We'll be studying also the developmental disorders, the few major ones which are relevant. So why is this topic important? So there is a famous saying actually, कि the architecture of mind is laid down before birth. So how you think, how you react, how you expect, your cognition, emotional intelligence, everything. So everything, the architecture, the foundational layout is laid down already before birth. So why does this topic matter? So as we all know that if brain, the architecture of brain is laid down before birth, so understanding the whole process will lead to its prognosis, its treatment, prevention of diseases, and further better analysis of the functioning of brain. At the end of the session, we'll be able to explain how the brain and spinal cord develops from the early embryonic life to the fetal life to the adult stage. Next, we'll be identifying the key stages in central nervous system formation, and also we'll be recognizing the major congenital and neurodevelopmental disorders of brain and spinal cord. So as we all know, what is central nervous system? So the basic definition, the basic thing what we all know, be it from the neuroscience background or anywhere. So the CNS comprises of brain and spinal cord. So there are two major parts of CNS. First is the brain, and after that the second is the spinal cord. And apart from these, the brain is protected by meninges, the cerebrospinal fluid which circulate in those meninges, as well as the primary protection, that is the bones, be it skull or be it the spine. So the embryological development. So after fertilization, the whole process starts. There is development of morula, there is development of blastula, which further differentiates into gastrula, and the whole gastrulation process takes place. So our story begins at the stage of morula. So in the morula stage, actually there are two layers. First is called the epiblast, the second is called the hypoblast. So the epiblast further forms the epidermal cells, mesodermal cells, and the endodermal cells. Our layer of interest is epidermal cell. So the epidermal cell, it forms in third week of gestation, and the CNS development process begins at the same time. Dorsal ectoderm dorsal ectoderm forms the neural plate, whereas the ventral ectoderm forms the peripheral nervous system. What is neural plate? So the neural plate is a keyhole type structure which is broad at the end and like broad at the anterior end, and it is narrow at the posterior end. So as we all know, the ectoderm develops from the epidermal cells. So the ectoderm, further it is of three types: the dorsal ectoderm, the ventral ectoderm, as well as the ventral edge. So it is the edgemost part ventrally. So the dorsal ectoderm lead to formation of the neural plate, which we'll be discussing subsequently. The ventral ectoderm leads to formation of peripheral nervous system, and the ventral edge cells. So there are some isolated ectodermal areas. Those areas are termed as placoids, and those areas give rise to sensory structures such as cranial sensory ganglions. So the whole process of the formation of the CNS, it is called neurulation, and it mainly comprises of 4 stages. So we'll be discussing all 4 stages subsequently. The stage 1 is termed as like the first stage or the primary stage. In this stage, the surface ectoderm, it actually differentiates and thickens at the center. So there are 2 different landmarks. First is the precordial plate, which forms the future mouth, and the second is primitive node, so that subsequently forms the cloacal area or the caudal area of the human being. So the area between precordial plate and the primitive node, it starts thickening, and that thickened area is called as surface ectoderm or neural plate. So this neural plate is also termed as medullary plate, and neural induction, that is formation of the neural cells, it caused by the suppression, it causes the suppression of epidermal fat. So after the epidermal cells are forming the neural cells, so there are few factors, few proteins, few factors which are responsible. So in that presence of that, like, it can lead to formation of ectodermal fat. So it can lead to formation of skin tissues, the exterior tissues, and if the factor is absent, it will lead to formation of neuronal cells. So that factor is termed as BMP, that is bone morphogenic protein. So as we discussed, the ectoderm isolated before gastrulation, the bone morphogenic protein, if it is present, it will lead to formation of ventral epidermal tissue. If BMP is absent, so it will lead to formation of neural tissues. And in case BMP was absent, neural tissue is formed, and after the formation of neural tube, BMP4 again comes into action. Then the neural tissue, the primary neural tissue formed, will be again further differentiating into epidermal tissue. So coming to the next stage, so that is called the neural folding stage. So in this stage, the neural plate, it starts folding in like M-like fashion. So there are two crests and one trough, that is one depression along the midline craniocaudally. So those two crests are termed as neural folds, that are the elevated margins, and the groove, the linear fold, that is termed as linear groove. And at the junction of neural plate and surface ectoderm, the cells differentiate to form the neural crest, and these cells of neural crest, they further migrate into different areas to form the peripheral nervous system as well as the autonomic nervous system. So in this diagram we can see clearly, the diagram A, it is of stage 1, where the thickened plate, neural plate, we can see clearly. So in the stage 2, that is diagram B, the neural folding starts, the neural crest and neural groove develops. In stage C, which we'll be discussing after this slide, after the folding, the upper layers start fusing together. So they will come close to both, and they will start eventually fusing in the craniocaudal direction. And stage 4 is formation of a hollow tube-like structure from cranio to caudal end, and this structure further leads to formation of vesicles as well as the spinal cord. So coming to the next stage, that is stage 3, that is formation of neural tube. So as we saw in the previous diagram, the neural fold, the two troughs, those come together, start getting closer and start fusing along the midline. And this fusion, there is a peculiar feature that it begins from craniocaudal direction. So that means it is in a zip-like manner. So when we are opening a zip, from top to bottom, so this fusion begin in the same style. And this fusion lead to formation of neural tube. So this neural tube further gives rise to our central nervous system, which we'll be discussing. So this fusing stage, as we can see on the day 22nd, the neural fold, the somites, otic placode, everything is visible. By day 23rd, the fusion is almost complete. So only two opening, that is the anterior neuropore at the top, the posterior neuropore at the bottom is visible. And coming to the last stage, that is neural tube closure. So as we saw in the last diagram, by day 23, there were only two openings left, that is the anterior neuropore and the posterior neuropore, which is called as cranial and caudal neuropore. So these opening, why is it left open? So the primary function of these opening is to provide nutrition. So these opening facilitate the circulation of amniotic fluids. So before the CSF circulation starts in our central nervous system, this amniotic fluid is the liquid, amniotic fluid is the substance which provide nutrition to all the neuroectodermal cell or to all the neuronal cell, we we can say. So yeah, these openings are left, and these opening, the cranial end, that is the anterior neuropore, it closes at the 25th day of intrauterine life, whereas the caudal end closes by the 27th day of intrauterine life. So this is the summary of what we discussed for the development of brain. So as we saw in the third week, the thickening of central part of ectoderm starts. There is formation of neural plate or medullary plate. Then in the second phase, the folding of neural plate starts, and the formation of neural fold, neural crest, neural troughs, everything develops. By the stage 3, this fusion, the neural folds, neural crests, they start coming closer and start fusing in craniocaudal direction. And the fourth stage, on 25th day, there is closure of cranial neuropore. And on the 27th day, there is closure of the caudal neuropore. And this whole process of 4 stages basically leads to formation of neural tube. Now what is neural tube? So it is a hollow tube that extends from our cranial end towards the caudal end, and this neural tube will further give rise to the brain as well as the spinal cord. So now the neural tube is formed. So the central cavity of neural tube, it forms the vesicles of brain and central canal of spinal cord. So as we all know, the brain consists of few vesicles. So there are 3 major ones, as we know, prosencephalon, mesencephalon, rhombencephalon, we might have heard of. So it leads to formation of them in the brain as well as the central canal in the spinal cord. And the peripheral wall of this neural tube form the tissues of the nervous system. The cranial cavity in the cranial cavity, or the cavity at the cranial end, dilates to form the brain vesicles, whereas the caudal part remains tubular throughout the life. So as we can see, the cranial part, it differentiates into 3 primary brain vesicles. The first is prosencephalon, the second is mesencephalon, and the third is rhombencephalon. And these 3 are further differentiated into 5 vesicles. That is, prosencephalon gives rise to 2 more vesicles, and rhombencephalon also 2 more brain vesicles. So yeah, prosencephalon forms the telencephalon, which further forms the 2 cranial hemispheres, and it also forms the diencephalon, which forms the deep brain structures such as optic vesicle, pineal, thalamus, hypothalamus, posterior hypophysis, and many more structures which are located in deep brain area. Second is mesencephalon, so it only gives rise to midbrain. And third is rhombencephalon. Again, it is divided into 2 major vesicles. First is the metencephalon, and the second one is the myelencephalon. So metencephalon further gives rise to pons, further gives rise to cerebellum, whereas myelencephalon, it gives rise to medulla oblongata. So as we can see in this diagram, by day 22nd, the 3 major vesicles, as what we discussed in the previous slides, the prosencephalon, the mesencephalon, and rhombencephalon, these 3 are prominent. By day 28, so apart from these 3 vesicles, we can also see 2 flexures. So what are flexures? Flexures, as the term signifies, they are the bendings. So first is the mesencephalic flexure. So it, as the name signifies, it lies near the mesencephalon. And second is the cervical flexure. So it lies caudally to the rhombencephalon and anteriorly to the spinal cord. So by day 30, there is one more flexure, that is pontine flexure. It forms in the middle. And by week 6 and week 7, the 3 vesicle further differentiates into 5 vesicles, and formation of the brain like proper occurs. Now coming to the derivatives of neural crest. So the neural crest, as we saw, migrates and forms different neural tissues. Few of them include neuroblast cell, which further gives rise to DRG, that is dorsal root ganglion, as well as sensory ganglion. Other is odontoblast cell, parafollicular cells of the thyroid, etc. The next group of cells which neural crest gives rise is spongioblast cells, and these cells form the satellite and the Schwann cells. And the third and also one of the most important group includes the pluripotent stem cells. And these cells further gives rise to melanocytes, which are responsible for melanin production. Now coming to the second part of our session, that is the spinal cord development. So as we saw, like the neural tube, the process of formation of neural tube, the process of formation of brain vesicles. In that vesicle, like the most posterior one or the most caudally one located was rhombencephalon. So the part of neural tube which is located caudally to the hindbrain, that is rhombencephalon, it forms the spinal cord. And this again occurs in 4 major stages, as we will be discussing. So the first stage is proliferation of neuroepithelial cells. So as we saw in development of brain, development of CNS primarily, the neuroepithelial cell differentiates and forms 3 layers here. So the innermost is ependymal cell layer, the middle layer is called the mantle layer, and outer layer is called the marginal layer. So here 2 layers will be our layers of interest, which will be further giving rise to several spinal cord structures, as we will be discussing eventually. So this mantle and marginal layers or mantle and marginal zones, so they start rapidly proliferating, and this proliferation lead to formation of 2 lamina. So those lamina are called basal lamina and alar lamina. So the alar lamina, it grows dorsally and it forms the spinal sensory nerves. So it is located posteriorly, the alar lamina, it forms the sensory tract, sensory pathways and sensory nerves overall. And it is separated by a structure known as dorsal medial septum. Apart from this, the basal lamina, it is the ventral structure. So it consists of motor nerves, motor pathways, and it is separated by ventral median fissure. So as we can see in this diagram, the pink one is the alar plate, which is located dorsally, and the blue one is the basal plate, that is motor in nature, and it is located ventrally. So here we can see 2 factors. So first is BMP, as we discussed, it stands for bone morphogenic protein, and it leads to formation of alar plate, that is sensory in nature. And the second one is SHH, and that stands for sonic hedgehog protein. So sonic hedgehog protein, if it is present, this factor induces the formation of basal plate, which is motor in nature. So presence of these 2 factors induces the formation of alar or the basal plate. And there is a structure which lies below the neural tube. So that structure is called notochord. So the notochord structure, it produces several enzymes, several factors, namely few of them includes nodine, choline, follistatin. So these factors, these regulate the action of BMP as well as SHH. The phase 3 includes the formation of different cells such as neuroblast, spongioblast, and their differentiation into neuronal cell layers. So this thing we already discussed, it is same as we discussed in brain. Phase 4, so after everything is formed, after the alar plate, basal plate is differentiated, by the 3rd month of intrauterine life, the cord extends throughout the length of vertebral canal. And after that, there are some positional changes, that is the cord starts lifting upwards. So at the time of birth, the cord extends to the L3 vertebra, and in the adult stage, it extends to the lower border of L1 or upper border of L2 vertebra. So this is a summary of the formation of spinal cord. Phase 1, it includes the formation of mantle and marginal layers. So there is proliferation of neuroepithelial cells, its differentiation, in which the innermost layer forms the ependymal cell, middle is the mantle layer, outer is the marginal layer. Phase 2 includes the formation of basal and alar lamina. So the basal lamina and alar lamina, as we know, they form by rapid growth of mantle and marginal layers, which are separated by sulcus limitans, and this rapid growth lead to formation of alar and basal lamina. Third part is histogenesis of the cell. So the neuroblast will form the neurons, the spongioblast will form the astrocytes, microglia, oligodendrocyte, and the mesoderm will form the microglial cells. And the phase 4, as we recently saw, there are some positional changes. At the time of third month, the spinal cord extends throughout the vertebral canal. At time of birth, it extends to L3. At the time of adult stage, it extends to the lower border of L1 or upper border of the L2 vertebra. Now coming to the last part of the session, so there are a lot of neural tube defects, like maybe some 50, 100 or even more. But here I have compiled like few of the major which we'll be discussing. So the first one includes the spina bifida. So as the name signifies, it leads to formation of bifid spines, and it majorly occurs within the first 4 weeks of the pregnancy. So this is due to incomplete closure of neural tube, and it is also a genetic disorder which results in underdeveloped vertebrae. So the vertebral column, it is not properly developed, which leads to like different degrees of severity, which can even be or not even be, which can in most of the cases it is lethal in nature. So the major 2 types include the spina bifida occulta, and the second one is spina bifida cystica. So in this, the first one, the spina bifida occulta, it is the least severe type, whereas the spina bifida cystica, it is again mainly of 2 types. So we can see in this diagram. So first is the occulta, it is the least severe, which leads to minor protrusion of the spinal cord to the vertebral column. So it is usually discovered, like we can easily see it on X-rays and scans, and people are mostly not even aware of this condition. The second, the meningocele and the myelomeningocele. So this 2 are the classification of spina bifida cystica. So there is a cyst-like formation in both of these. In the second one, as we see in meningocele, so it is a serious condition. So in this, as the name signify, there is protruding meninges outside the vertebral column. So this can be seen like a cyst-like swelling, and often usually visible through MRI or even at CT scans in certain stages. The third is the most severe form, that is also called the myelomeningocele. So why it is called myelomeningocele? So apart from the meninges, the spinal cord also starts protruding outside the bulge, outside the cyst, and this is the most serious type, and the spinal cord is also enclosed in the cyst. The next type of developmental disorder includes anencephaly. So anencephaly, as the name signifies, it is non-proper development of the cranial cavity, of the covering of the brain. So in the first diagram we can see the brain is underdeveloped, and the skull line is also not properly developed. And the second diagram we can see the comparison with a fully developed and normal child. The third is encephalocele. So this is a condition which is caused due to non-proper closure of the occipital bone. So the occipital area, the occipital bone of the cranial cavity does not closes properly, which further leads to protrusion of the brain, protrusion of the cerebrum outside, and this condition is always, most of the cases, usually very severe and lethal. Next is anencephaly. So it also involves a defective occipital bone, and in this condition there is a retroflexion of the head. So the head is bent backwards due to defective occipital bone as well as spina bifida of the first cervical vertebrae. Next is lissencephaly. So in this, like as we all know, there are structures called gyruses and sulcus. So gyruses and sulcuses are present throughout the cortex of brain, and these convolutions are not formed in this condition, that is lissencephaly. So failure in development of convolutions in cerebral cortex due to defective neuroblast migration, whereas the deep brain structures, like those are not affected and they remain normal. So coming to the summary of the whole session, neuroectoderm forms neuroectoderm forms from the ectoderm during gastrulation, and this further gives rise to CNS. The neural plate forms and folds to form the neural tube, which is also called neurulation, that occurs around week 3 to 4. The neural tube further becomes the brain, which is located cranially, and the spinal cord, which is located caudally. And the neural crest cells, they migrate to form parts of peripheral nervous system, and the brain formed after the neurulation process, it further differentiates to form primary vesicles, 3 primary vesicles, that is forebrain, midbrain, hindbrain, also known as prosencephalon, mesencephalon and rhombencephalon. So this primary vesicle further differentiates into 5 secondary vesicles, and as we saw, and these 5 secondary vesicles further forms the major structure of the brain, including the cortex, thalamus, midbrain, pons, medulla, and all deep brain structures. The spinal cord develops from the caudal neural tube, and motor neurons from the basal plate arise ventrally, and the sensory neurons from the LR plate arise dorsally. And the process of myelination, it starts before birth, it continues postnatally, which helps in efficient nerve conduction. So these are my references. Thank you guys.